Today you won't be seeing me, just the presentation. I'll be discussing the interorgan flow of amino acids and the metabolism of amino acids and specific or select organs that I've decided to talk about. So let's start with the plasma. After you eat, the plasma concentrations of amino acids increase or rise. We keep a pool of about 150 grams of endogenous and exogenous amino acids. And we think that they're reused, so the primary source of our amino acids that we need for protein synthesis is through reuse. We recycle. And there's more non-essential amino acids than essential amino acids kept in the pool, which makes sense. In terms of interorgan flow, let's consider enterocytes, so small intestine cells. The amino acids are used for structural proteins in the cells, nucleotides. So that's the DNA. Apoproteins necessary for the formation of, for example, chylomicrons, making new digestive enzymes, hormones, and other nitrogen-containing compounds. I want to talk about glutamine because if you ever work in a hospital setting and you have a patient in the ICU, glutamine is sometimes, or often, we give a formula, a tube feeding formula with glutamine, or we even may give a supplement that has glutamine in it. It's a source of energy, but more importantly, it's trophic, meaning it causes or stimulates the growth of the gastrointestinal cells. And even more important, it prevents bacterial translocation. What does that mean? That means the gut that's in the gut that's in the intestine, we don't want going anywhere. It needs to stay in the intestine. When people get very sick, and I'm talking ICU level, if they have sepsis, the bacteria tends to migrate. There tends to be translocation of the bacteria into the systemic circulation, and that's a big problem. You can get glutamine from your diet, as I was saying, or it can be metabolized. It enters the portal blood and it ends up in the liver. And you can make glutathione, proline, or ornithine from glutamine, and very little leaves the intestines in a good state of health. When we're talking about glutamine in the liver and kidneys, we need to be aware that ammonia transport is a major role of glutamine. Extrahepatic tissues can catabolize excess ammonia to make glutamine. It can be released to the muscle, to other tissues to provide energy. And as I was saying, during periods of trauma, the use increases dramatically. I gave you the example of sepsis, which is a system-wide infection where you might want to administer glutamine in a patient who can't eat via tube feeding, or actually give them a supplement with glutamine in it. Skeletal muscles use amino acids, of course. They have a preference. They like aspartate, asparagine, glutamate, isoleucine, leucine, and valine. And we do have nitrogen-containing compounds that act as indicators of muscle mass and protein catabolism. In acute periods of stress, there is profound loss of muscle tissue to the point 20, 30 pounds, depending on the original weight of the individual. I had a associate who was in a young, healthy male, my version of young, early 30s, young, healthy male, ski accident, ended up in the ICU, profound weight loss. And this person may have gone, we don't go any longer, 5 to 7 days without some source of nutrition. So tube feeding or total parenteral nutrition, you don't lose in 5 to 7 days, you don't lose 20 to 30 pounds. So this acute response to stress with this increased metabolism causes this weight loss, and unfortunately most of it is skeletal tissue. In the kidneys, when we talk about amino acid metabolism, we have glutamine catabolism to maintain acid-base balance, and that includes glycine as well. There are a number of reactions that occur. We talked about arginine synthesis from citrulline. We just talked about tyrosine synthesis from phenylalanine. And we also have histidine synthesis from carnosine degradation. So each organ kind of has their own thing going on, their own specialty, if you will. It's kind of a good way to think about it. The brain, that's pretty important in accessory tissues. So glycine is an inhibitory neurotransmitter, so it puts the brakes on, and that's in the spinal cord. Taurine, the one that's important for cats as well, acts as also an inhibitory neurotransmitter. Aspartate has the opposite effect. It's thought to be an excitatory neurotransmitter, and that's in the central nervous system. And glutamate acts primarily on the brain and spinal cord as an excitatory neurotransmitter as well. Biogenic amines, so we're talking about those amino acids that stimulate some of the feel-good hormones. Tryptophan, we sometimes are familiar with after we eat our turkey. We kind of get that kind of content feeling, and that's said to be from the synthesis of serotonin. We have the fight or flight hormones. We have dopamine, which is a feel-good one as well, from tyrosine and norepinephrine and epinephrine. And histidine is decarboxylated to be generated. Neuropeptides, as I was saying, we're going to talk about now neuropeptides, and what they are small protein-like compounds, and they're very similar to neurotransmitters, but they have more diverse effects than neurotransmitters do. So they have impact on hormone releasing factors, they have endocrine influence, they can modulate transmitter function, mood or behavior, and they actually have neurosecretory cells, or we have them in the hypothalamus, which secrete these neuropeptides. And that concludes my lecture. Thank you.